Every day in the UK, 2 to 3 people become paralysed as a result of a spinal cord injury. That's one person every 8 hours. Of those, they're typically young, aged between 15 and 30. That adds approximately 1,000 people to the 40,000 people that are already living with paralysis. I was aged 22 when I broke my neck playing rugby for a season in New Zealand, and that was over 20 years ago. I can recall back then that discussion and hope in relation to treatments or a cure was very much discouraged, and for the first 10 years things appeared to move very slowly. Over the past 10 years, however, things have made remarkable progress, and the past 5 years in particular have accelerated dramatically. Progress thus far has been made with very limited financial resources. Spinal cord injury in the UK does not receive any funding from government and is typically dependent on charities. The limited opportunities for funding led me to embark on a challenge dubbed the Mighty Push in April last year to raise money for research and also to mark the 20th anniversary of my injury. As with most crazy plans, it started on a night out. The night started pretty well, with a few beers. However, it ended with me having to hand cycle 250 miles around New Zealand. We did a section from Christchurch, which was 100 miles, and then round Lake Tekapo, which was another 50 miles, and the last section round near Dunedin, which was another 100 miles. One of the things that made the challenge so special for me was that so many family and friends came out to join me on various sections. Some, however, got a bit carried away with the excitement of it all, taking the cycling theme far too seriously. There's a lot of extremely promising lines of research already identified, and I feel strongly that we need to push harder to get them as quickly as possible into human applications. And this seems an appropriate point to introduce Professor Keirstead. Professor Keirstead completed postdoctoral studies in Cambridge University in England before going to the United States, where he is now Professor of the Reeve Irwin Research Center at the University of California, where he also founded the Stem Cell Research Center. He directs a research team investigating the cellular biology and treatment of spinal cord trauma, research that also has significance for other diseases of the nervous system. And I'll now hand over to Professor Keirstead to talk more about the developments in spinal cord injury.
I began working in spinal cord injury, which over time moves from acute to subacute, days or weeks after the trauma, to chronic, over weeks and decades. Each of those stages of spinal cord injury should be regarded as a completely different injury. Early on, acute spinal cord injury is characterized by gross inflammation. Your immune system rushes into the area of damage and makes it worse. Then after a few days, that inflammation dies down and you just have a cellular deficit, a hole. That's subacute chronic spinal cord injury. And then after time, one has the accumulation of a new cell type, a scar. And all three stages of injury require completely different approaches because the target for therapy is completely different and the barriers to therapy are completely different. So I began breaking them up and began working in acute spinal cord injury, and actually didn't use a stem cell approach. So I'm just going to tell you about that very briefly, as this was really my education as to how to take something from the bench to the bedside. This treatment that I developed with my team at the Reeve Irvine Research Center is now in phase 3 clinical testing with Bristol Myers Squibb. So if you imagine that this is a blood vessel and inside it is the blood in here, in the middle of the blood vessel called the lumen, if there's an injury out to the side, the cells get pulled out, they get recruited from the bloodstream out to the site of injury by particular recruitment molecules. What I did was developed a way to stop those recruitment molecules using an antibody, a little protein that binds them up. So what we did was we injured mouse spinal cords using a 0.6 millimeter blade, very, very thin blade, cutting 50% of their spinal cord. But you can see here 2 weeks after the injury, that is not a 0.6 millimeter injury. It has now extended several millimeters towards the head and the feet. You can see that less than half of the spinal cord is now still there, and you've lost tremendous amounts of tissue towards the head and the feet. Tissue that otherwise would have had function. But if we get in there within 6 hours of injury and inject anywhere in the body, into the muscle, peritoneal cavity, into the bloodstream, an antibody, a small molecule, or small protein rather, that binds up and renders non-functional the recruitment signals from the site of injury, we don't get a particular component of the immune system rushing into the injury site. We've bound up the recruitment molecule. And then what we see 14 days later after the same injury, we can't even tell that the cut was there because the skin or meninges of the spinal cord's grown over. But if you cut that spinal cord in half and take a look at it from the side, you can still see the original cut site. But look at the amount of tissue that is spared. 70% of the tissue that would have been lost is now preserved. If you appreciate that it only takes 5 to 10% of a spinal cord to allow somebody to walk, it's quite tremendous to be able to spare 70% of the tissue that would otherwise have been lost. When we look at the movement of these mice running over a glass tabletop, now we're filming it from underneath, you can see that this mouse variously drags or hops. It's had 50% of its spinal cord cut with no treatment. It can move a little bit, but very, very little. The same injury, if we get in there within hours of the injury with a simple needle injection, we see this in every case. This is not an outstanding example. Full coordinated weight-supported locomotion. We slow these videos down and measure such things as joint angle and toe spread and stride length and stride width and all this type of fancy stuff, but you don't need to see all that data. Suffices to say that we have largely attenuated the secondary damage that's caused by CD4-Th1 T cells, or a component of the immune system, from rushing in and making the spinal cord injury worse. It costs roughly $1 billion American for one treatment to make it through. Now that includes the cost of money, and it also includes the failures, but it's roughly $1 billion in order to get one treatment working through the clinical trials of phase 1, phase 2, and phase 3 testing in humans prior to marketing. Large companies need about $1 billion a year profit in order to pay for all that stuff. And spinal cord injury at 100% market penetration, meaning that if one of us were to invent a drug that worked in every single spinal cord injured patient that took it, it's only a $300 million a year annual market in the United States. So knowing that, I then embarked upon, with partners and colleagues at the university and companies, an approach to show that this same treatment also halted multiple sclerosis, multi-billion-dollar market, and rheumatoid arthritis. There's the biggie, $6 billion annual market. Now, you may think it odd that one single drug could halt spinal cord injury, secondary damage, MS, rheumatoid arthritis. What I'm not showing you is ulcerative colitis, Crohn's disease being a component of that, and of all things, herpes infection of the eye. So we showed in 3 out of 3 of the pharma-accepted animal models of multiple sclerosis that we completely halted multiple sclerosis. It's actually the only drug to go through preclinical testing to have done that. We cumulatively made the market size about $11 billion annually, prospectively, if this was to work in humans. Now we got attention, and I'm really pleased to say that Bristol Myers Squibb ended up taking this program. So after tens of thousands of dollars startup money, they have now poured in undoubtedly hundreds of millions of dollars at this point. We actually ended up making the decision with Ability Biomedical, then Medarex Corporation, then Bristol Myers Squibb, to go for indications that were inexpensive and fast with easy outcome measures. And then the idea is to march it through the clinical testing and then use it off-label for spinal cord injury and multiple sclerosis. I'm most excited personally for spinal cord injury, my field of interest, but it has to be administered within hours of a spinal cord injury. With MS, anytime during the course of the disease. Same thing with ulcerative colitis or rheumatoid arthritis. But in spinal cord injury, this molecule is only released for a brief period of time, 12 hours in a human. So you've got to get in there. But you can. Most emergency rooms see patients within 45 minutes in first world nations. So very conceivable to administer this in a single injection. And if I was to predict something, albeit with a little bit of hope, I think that this drug is going to be administered to every CNS trauma patient or disease upon a manifestation to an emergency room. So after it got into the clinic, I left it and I watched like you do, and then I moved on to subacute spinal cord injury. And just a few words about this. Now we've got a situation where the inflammation is over largely, and we just simply have a cellular deficit. So what I did with my team at the Reeve Irvine Research Center at UCI, we developed a cocktail, a series of cocktails actually, that mimicked human development. So we looked back at human development when this particular spinal cord cell type was being made. What growth factors, which hormones, which substrates, what pH levels are existent in the human during the development of this particular cell type. The tough part was, at any particular stage of human development, there's a whole bunch of things going on. So to sort out which ones were specific for our cells and not others was difficult. What we did is we developed a procedure where we could take one single stem cell and make it into 11 oligodendrocyte progenitors. Young cells of the spinal cord that I had theorized were responsible largely for the damage and lack of locomotor abilities, and therefore may be responsible for repair. Almost every human injury, there's a region of spared tissue around the outside of the hole or damage. And if you imagine that these are your brain cells or brain stem cells whipping down your spinal cord, they carry electricity because they're wrapped like a wire in a wall with insulator called myelin. And I had theorized that that myelin deficit was responsible for rendering these otherwise intact axons from conveying electricity. So that's why I took stem cells, made them into oligodendrocytes, the insulator producing cell type, in order to transplant them, show that it reinvested those wires that were spared but otherwise non-functional, and rendered them functional again. This was the first human embryonic stem cell-based clinical trial in the world, so there was no precedent. We couldn't go to the FDA, nor could we go to any medical doctor and say, what's the clinical plan for our cells? We had to gather people together and discuss risk, patient inclusion criteria, patient exclusion criteria. But now that it's done, I'm really, really pleased to say that Geron stuck in there, has consistently renewed their funding and support for this, a terrific group of individuals, and we were approved. I began working with stem cells to generate another cell population, and this was my stepping stone and major breakthrough to that cell population, making brain cells. This is neural. In your brain, you have two large classes of cells called glial cells that support, and neurons that are actually your primary brain cells, if you will. Some of the glial cells make scar tissue, so transplanting them into sites of trauma is problematic or risky. So I decided that I would try to move towards a very, very high purity population of particular brain cells, and this was the stepping stone, and I'm really excited about this. I pushed these cells to become motor neurons, my intent. Motor neurons are those cells that seed your spinal cord. Your brain connects to them, and then the motor neurons then project out to every moving thing in your body. Your muscles, yes, but remember also your sphincters, your heart, your lungs, every moving thing in you is controlled by a motor neuron. We've now generated what I believe to be the world's highest purity of motor neurons. We're at about 99.6% pure. They're functional. We can incubate them with human muscle, as you see on the right side of the screen. And without that motor neuron population being there, the muscle is quiescent and still. When we allow the axons to grow in, this muscle is from a 67-year-old man, we get beautiful muscular contractions. So de facto, we know that our motor neurons are real. Our motor neurons are doing two things. In some models, they can actually project and hook up to muscle. In other animal models, they just secrete growth factors that keep the endogenous motor neurons from dying. So we can save muscle mass. California Stem Cell has generated a very complex manufacturing system, so we can now produce the cells in a manner that's clinically viable. It's completed a GLP, or FDA compliant, safety study, and we expect in about a year we'll be in the clinic, maybe 18 months at the outside. This should be the first motor neuron stem cell-based clinical trial in the world. Perhaps the second stem cell-based, human embryonic stem cell-based clinical trial. It really is the dawn of a new era. I know that sounds corny, but it's not. We are truly at a tipping point right now in medical science, and certainly in spinal cord injury, where we are beginning to see that transition from hope and pontification of possibility into true application. It's a fun time. Thank you very much.
I found it just inspirational.
Really interesting and so quite sort of encouraging.
What it offers to medical science in general, I thought was fantastic.
It was very illuminating, informative and pretty exciting.
Well, David, as you know, is inspirational, and to do 250 miles, which was in 10 days.
I think it just shows the determination that he has is quite incredible.
It's generated an amazing amount of interest and support, and I just really hope that we can harness that and take it forward.
These things all need money, need huge funding, and that's what we're hoping for.
Nowadays, with funding levels as difficult as they are, one needs more early stage proof of concept, and that is really the role filled by the donor community.
I think one of the really nice things about Hans Keirstead is that he can present very complicated scientific information in a way that's understandable to everyone.
My research has always been very, very applied, and I think that attracted David to my group, and I was very attracted to his team because they were very, very directed at getting applications into the field.
It's heartening to hear that there's so much development in science taking place, and that there's a potential light at the end of the tunnel for the treatment of catastrophic neck injuries.
It's not only rugby, it's sports, it's crash accidents. If this solution can be universal, it's going to be much greater than what we thought.